Okay, now that you've got all your homologous pairs together, now you're going to line them up. Again, this is separately, so the people that have the blue sheets, separate from the people that have the pink sheet. You're going to imagine your desk, or the area in front of you, is your cell, and you have all these groups of homologous chromosomes lined up or set up on your desk. So they're all paired up in their homologous pair. What you're going to do in your cell is then put them down the middle or the equator of the cell. All right, so, or imagining your desk in front of you is your cell, so your chromosomes are going to line up in their homologous pair. So I'm just going to do numbers. So let's say chromosome 2 with its homologous pair 2 next to it. Chromosome 14 with its homologous pair 14 next to it. Chromosome 5 with its pair 5 next to it. Chromosome X with its pair Y next to it. So all of these represent 2 chromosomes lined up down the middle, but each of those chromosome sets is a homologous pair. The other thing I want to point out to you at this point is I just randomly chose this order, 2, 14, 5, X, Y. The way the homologous pairs line up at this point is completely random. All right, it doesn't have to be 1, 2, 3, 4, 5 all the way down to the 23rd pair. They just get randomly moved to the middle of the cell and lined up in whatever order they happen to be. They also are randomly lined up, if you look around at the people who are lining these up, as to where the father's chromosomes end up and where the mother's chromosomes end up. Okay, so again, if this is a male and his testes are going through meiosis, the chromosomes he got from his mom or his dad line up randomly on either side of this imaginary equator here. Here's the middle of where they line up. So they could have lined up so that all of the mother's chromosomes were over here and all the father's chromosomes were over here. So we'd have all pink or blue, or pink or red over here. We'd have all blue over here. Or in the case of the male, all orange on one side and all purple on the other side. They may have lined up that way, but it doesn't have to be. Okay, the mothers too could line up on the left side or the right side. The mother's 14 could have lined up on the left side or the right side. It's completely random what order they line up in and which side the homologous pairs line up on. Completely random. All right, if you don't already have them lined up down the middle of your desk, please do that. You might have to do it the long way of the desk, depending on how long your chain of chromosomes are, to kind of spread out and find a space for lining up those homologous pairs. If you're not done yet, you want to pause it now before I move on. So we had the homologous pairs all lined up down both sides of the cell equator. Again, imaginary line through the cell. I'm just going to show all the chromosomes as X's now instead of numbers so that we can keep track of what's going on a little bit more quickly. So you're going to have 23 pairs. Again, I'm not going to draw them all, but 23 of these chromosomes lined up down the equator of the cell. And the first division of meiosis happens, which means all of these chromosomes get divided up from all of these chromosomes. So they split in their opposite ways. Okay? So some go to this side of the cell, this group right here, and some go to this side of the cell, this group right there. So we divide the homologous chromosomes. So I want you to do that. Okay? The right side of your cell, those chromosomes in that row need to go pull to the right, and the ones on the left need to pull to the left. They don't have to have a huge amount of space between them, but just separate them enough so you can see a distinction between those different sets. All right, moving on then. What you should have when you're all done is 2 rows of chromosomes pulled to opposite sides of your desk. Again, we're still in one cell, but they've divided in half in that cell. If you remember from the vodcast, the little pictures I showed of the mouse sperm being produced, okay, this is after that first division. We just see 2 dark clumps of chromosomes on either side of the cell. Okay, now we're going on to division 2, or meiosis 2. What happens in meiosis 2 is the sister chromatids split apart. Remember that in each one of these X's, okay, is actually a copy of DNA, two copies of DNA. Right? If we zoom in on one of those X's, here is one version of the DNA, and here's the other version, the copy of the DNA, held together by a little centromere. Okay, these two parts, these two copies, are called the sister chromatids. So what we're going to do, as I said in meiosis 2, or the second division, is divide those sister chromatids. Now this may seem a little tedious at this point, but it is important to understanding how meiosis works. You are going to divide your sister chromatids by cutting each of your chromosomes in half. Now first of all, before you do that, I want to explain what I mean by cutting them all in half. So if we go back to one of these pictures, so if you have your chromosomes cut out already, okay, these each represent a set of two sister chromatids, so it's double the DNA. Normally what I would look for in a real chromosome is they'd have to get split in half the longitudinal way or the long way, so it'd be like splitting this one that way. Now, we don't have time to cut all of your chromosomes in half the long way. That's just, as I said, too tedious. But you can quickly snip them horizontally, just to show they've been cut in half. So for each of your chromosomes on each of your sides, you're just going to snip them horizontally to show that the sister chromatids have split in half. And now we've got two sets of genetic material for each set of sister chromatids. Okay, so you're going to go through and snip them each in half. Don't do it yet, because when you're snipping them in half, I want to make sure that you're staying organized. Again, I'm going to draw my cell again one more time. Here's your cell. Okay, here are your two sets of homologous pairs that have divided from each other. Okay, we're going to snip the sister chromatids in half, as I said, the horizontal way. Okay, and then one of each half is going to get pulled apart. So one half is going to go this way, one half is going to go that way, one half this way, one half that way, so forth and so on. Then you're going to snip the other side in half. And then one half is going to go this way, and one half is going to go that way. Make sure you keep these two apart, though. What we want to end up with is two rows, essentially, of sister chromatids. So I'm just going to do these in a different color so they show up better. But one half is going to end up over here, one here, one here, one here. So we're making two rows on this side for these chromosomes. But the chromosomes on the other side also get split in half into two rows as well. So now, as I said, we essentially have a total of 4 different sets of chromosomes. So when you're all done, you're going to have 1, 2, 3, 4 sets of chromosomes. This equals the 4 cells that are made in meiosis. Now those cells, once you get done with them, you're going to go back and you're going to count. Each of them should have half the number of chromosomes in them that we started with. So 23 chromosomes in each cell, because we started with a total of 46, or 23 pairs. So why don't you go ahead and do that if you haven't already finished. Snip them each in half, divide them in half, end up with 4 different groups of chromosomes, and you can actually put them in a pile that represent the 4 cells. Now the last thing is to look at what we ended up with in each of those 4 cells. These 4 cells represent, remember, the sperm that's being produced if you had a blue sheet, or the eggs that are being produced if you had a pink sheet. So we're curious as to what types of traits were passed on to those different cells or gametes. So I'm going to make my 4 cells again. 1, 2, 3, 4. If you had a female, so you start with a pink sheet, and she was producing eggs, in every single one of your cells, you're going to end up with X's. So she can only pass on X's. If you had a male, in other words, you started with a blue sheet, in 2 of his cells, he ended up with X's, but in the other 2 cells, he ended up with Y's. So, if the people with the pink sheet, the eggs, okay, if one of their little eggs would end up joining with one of these 2 sperm cells, the result would be an X from the egg, a Y from the sperm cell, creating a son. If the people from the pink sheet, again the eggs, ended up joining with these 2 sperm cells, either one of them, the result would be an XY, or sorry, an XX, in which case it would be a daughter that's born. So in reality, whenever a couple has a child, they have a 50-50 shot at having a son or a daughter, because out of every 4 sperm cells that are made, 2 of them carry an X chromosome and 2 of them carry a Y chromosome. This also works for other traits, okay, not just whether you're male or female, but other genes as well. So I just want to give you an example. Make this a little bigger, erase faster. I'm going to give you an example of other traits that might be inherited this way. Let's say that the mother, the people with the pink sheet, had in all of her eggs, so in the 4 eggs that are produced, and every time she continues to make eggs, she carries the gene for blue eyes. So all of her chromosomes that she donates to her babies, and let's say this is on chromosome number 1, so all of her chromosome number 1s in those cells for those eggs are going to be carrying a blue-eyed gene. But the dad, and his chromosome number 1, he's got from his mom green eyes, and from his dad brown eyes. Okay, so chromosome number 1, we're not talking about X or Y here, carries the trait for eye color. The mother can only give blue eyes, but the dad could give green or brown. So depending on what combination we have, when the egg is fertilized by the sperm, that determines what the offspring are going to have for their traits. And again, the way they line up in meiosis and how they divide out in the gametes determines what those traits are going to be.